Welcome to part 3 of this video on the immune system. Okay, so we're currently in the process of discussing the acute inflammatory response, which is, remember, the second line of defense and part of the innate immune system. So it's a non-specific response, but it's the response that we initiate once a pathogen has actually breached the first line of defense. So the pathogen is now present within one of the tissues of our body, and we now want to see how we're going to eradicate it from that tissue of the body. So far what we've seen is that it will be recognized by sentinel cells as being there, and those sentinel cells will start to release alarm molecules. And these alarm signals are known as pro-inflammatory mediators, and we've seen 6 examples here. So tumor necrosis factor, interleukin 1 and interleukin 6, these are what are known as the pro-inflammatory cytokines. Prostaglandin I2, prostaglandin E2, these are the prostaglandins, and all of those 5 are going to be released by all 3 of the major types of sentinel cells. Then we had histamine, which was just released by mast cells. Now what I want to do is actually see what these pro-inflammatory mediators are going to do. And in the previous video I told you that the acute inflammatory response, fundamentally it's a response of the microvasculature. Remember what we're trying to do, we're trying to recruit troops in the form of proteins and cells, white blood cells, from the blood into the extracellular fluid. And fundamentally, how is that going to occur? Well, it has to occur by changes in the microvasculature. So really, the acute inflammatory response is a response of these microvasculature blood vessels to these pro-inflammatory mediators. And what I want to now understand is what is the response going to be of the microvasculature to these pro-inflammatory mediators. Okay, so where are we going to begin then? We're going to begin with histamine. Then we're going to talk about tumor necrosis factor alpha and interleukin 1, and we're also going to be talking meanwhile about prostaglandin I2. Now the reason we're going to do it in this order is histamine is going to be the one that's released first, because remember, histamine is already produced and stored in those granules inside mast cells. So the instant the mast cell gets activated by a pattern recognition receptor, it can just release those histamine granules, a process known as mast cell degranulation. So that's an important term here. Degranulation means the mast cells releasing their histamine granules. And that can occur extremely quickly. Whereas these other pro-inflammatory mediators, they actually have to be produced, they're not stored. So tumor necrosis factor alpha and interleukin 1, they're going to have to be produced, which will require protein synthesis. So will interleukin 6. Prostaglandin I2 and prostaglandin E2, they're also going to have to be synthesized from arachidonic acid, which again will take time. It'll be quicker than the production of proteins, but still it won't be as quick as just degranulating preformed granules of histamine. So this process is going to occur, this one's going to be released first, and then these ones will come later. So the ones that I want to discuss first, because they're the most important, are histamine, tumor necrosis factor alpha and interleukin 1, and we'll see that prostaglandin I2 is central to the mechanisms of those, so I'll just add it on as a side along. Then what we'll do is come back to talking about interleukin 6 and prostaglandin E2, because they have slightly different effects. Interleukin 6 is responsible for something known as the acute phase response and also causing a fever. Prostaglandin E2 is responsible for pain. It's very important in pain in the inflamed area. Okay, so we'll come back to those. So, what I'm going to begin with then is histamine and the effect that histamine is going to have on the microvasculature. Okay, so histamine is going to act on the endothelial cells of the microvasculature. So it's been released by our mast cells, it's now gone into the extracellular fluid, and it will now be able to diffuse around to these microvasculature blood vessels, and it will be able to bind to receptors that are on the basolateral sides of the endothelial cells of these microvasculature blood vessels. So let me just now draw an endothelial cell here. So here is an endothelial cell, much larger than on these previous pictures. And now on the basolateral surface of these endothelial cells, which is the side that the histamine is going to have access to, because remember the apical side faces into the lumen, the histamine is only going to have access to the basolateral side. On the basolateral side of these endothelial cells then, you're going to have receptors for histamine. And these are G protein coupled receptors, hence why I've drawn the classical 7 membrane spanning alpha helices structure here. And this receptor is specifically known as the histamine type 1 receptor, the H1 receptor. So I'll just colour this receptor in red here. So the histamine is going to diffuse over to these endothelial cells of the microvasculature, and it's going to activate H1 receptors on their surface. So here, the histamine is going to activate these, and this will activate an intracellular signaling cascade that the details of which I'm not going to go into, but it is GQ coupled if you do want to follow up on that. So it's going to activate intracellular signaling cascades, and this is going to lead to a process that is known as type 1 activation of endothelial cells. So histamine causes a type of activation of endothelial cells that is called type 1 activation. Now we'll see that the other really important pro-inflammatory mediators, tumor necrosis factor alpha and interleukin-1, they're going to cause type 2 activation in endothelial cells, which will be slightly different. So type 1 activation is going to occur because of histamine, and type 1 activation is an extremely quick type of activation. The intracellular cascade will occur very quickly, and the changes in the endothelial cell that are brought about will occur very quickly, whereas type 2 activation is much slower. Okay, so what actually is going to change then about the endothelial cell? How is the behavior of this endothelial cell that has undergone type 1 activation going to change? Well, there are 3 major components here. Firstly, it's going to start synthesizing prostaglandin I2. So type 1 activation causes the endothelial cells of the microvasculature to start synthesizing prostaglandin I2. Just like the sentinel cells were producing prostaglandin I2, histamine is now going to activate the endothelial cells also to produce prostaglandin I2. This is what I mean when I say that prostaglandin I2 is central to the way in which histamine and tumor necrosis factor alpha and interleukin-1 actually act. So it being produced by the sentinel cells will just merge into my discussion here. Okay, so these endothelial cells are going to now start producing prostaglandin I2. Now, what's the significance of the prostaglandin I2? Well, prostaglandin I2 has effects on vascular smooth muscle cells. So the prostaglandin I2 that's released by these endothelial cells is going to go into the extracellular fluid, and the idea is it will diffuse over to the arteriole here, where we've got vascular smooth muscle cells surrounding the arteriole, and it's going to act on the vascular smooth muscle cells, and it causes relaxation. So I'll just put this up here. So prostaglandin I2 or prostacyclin causes relaxation of the vascular smooth muscle cells, and therefore vasodilatation of the terminal arterioles. So it's going to lead to vasodilation, or if you really like, vasodilatation, but vasodilation is shorter, so we'll go with that word, which therefore is going to increase the lumen size of the terminal arteriole, and therefore increase the amount of blood that's flowing through the terminal arteriole. So it's going to increase the amount of blood that is flowing through your capillary beds in the tissue, and therefore you're going to get increased blood flow to that area of tissue. So this is going to raise blood flow. Now, why is that a good idea? Well, remember the big picture. What are we trying to do? Recruit troops to our area. If we've got a higher blood flow, then we've got more troops flowing through that area at any one time, and therefore we've got more chances to recruit troops out of the blood into the extracellular fluid. So increasing blood flow sounds like a good idea. It sounds that it's going to help us get troops into the extracellular fluid to fight the pathogen. Okay, so prostaglandin I2 is going to help increase blood flow. So the prostaglandin I2 up here that was produced by the sentinel cells is going to do exactly the same thing as the prostaglandin I2 that's now been produced by the endothelial cells in response to histamine. So we've now covered what prostaglandin I2 is going to do. So histamine causes type 1 activation in endothelial cells. This is going to cause them to release prostaglandin I2, and it's not just the endothelial cells of the terminal arteriole that will release prostaglandin I2. All of the endothelial cells will start releasing prostaglandin I2, and it will diffuse over to the arterioles and act on the vascular smooth muscle cells there to cause them to relax down, and therefore dilating the lumen of the terminal arterioles to increase blood flow. Okay, so that's part of type 1 activation. That's the first part of it. Secondly, histamine is also going to cause a process known as endothelial contraction, and this is a really important concept. So what's going to happen is the endothelial cells are going to contract down a little bit and open up gaps between them and their neighbours. So we're going to get this process known as endothelial contraction. So let me just draw a little picture for this. So let's say we've got our endothelium here. Let's say we've got our basement membrane here. Oh, and it's important to note here that the endothelial contraction is majorly going to occur in the capillaries and the post-capillary venules rather than in the terminal arterioles. So this majorly affects the capillaries and post-capillary venules over the terminal arterioles. And I'll explain why it's better to have it occurring in these than these ones in just a moment. Okay, right. So let me now just draw some endothelial cells on here. So normally the endothelial cells are like this. So here are two endothelial cells, and you can see that in the junction between the endothelial cells there isn't that much of a gap, and indeed they have tight junctions between them as well. So the gap between them is sealed, and this, you know, in the case of the capillaries, this will allow oxygen and carbon dioxide to diffuse across, and also a bit of glucose, but it won't allow huge proteins from within the blood plasma to actually leave the blood and go into the extracellular fluid. So what's now going to happen as part of type 1 activation is the endothelial cells are going to contract down, and in this process they will recoil their lateral edges like so. So they're going to go from looking like this to looking more maybe like this, okay? Where they've got larger gaps between themselves and their neighbours. They're going to open up gaps between the endothelial cells in this way. So the endothelial cells are going to be activated to contract down and open up gaps between them. Now, why is this a good idea? Well, this is going to occur in the capillaries and post-capillary venules, and you can imagine that what will then happen is fluid from the blood will leave through these gaps and go into the extracellular fluid. And this is going to bring in all sorts of proteins from the blood, which will then be able to fight infection. So this is going to result in the formation of what is known as an inflammatory exudate. So an inflammatory exudate is the fluid that is produced when the endothelial cells of the capillaries and post-capillary venules have contracted down in this way and opened up these gaps. It's the fluid that will be produced in the infected area, and it contains loads of proteins from the bloodstream that will be able to attack pathogens, principally the complement proteins, and we will go over the complement proteins later on. I just want to complete one story at a time. We'll cross one bridge at a time. We'll come on to why forming an exudate is such a good idea later on, but at the moment just know we are bringing in troops that are going to help kill the pathogen. Okay, so you bring in fluid that contains a huge number of proteins, so it's very important that this isn't just fluid. This is fluid that contains proteins from the blood that wouldn't usually be able to cross the endothelium of the capillaries and post-capillary venules, but which now can, and which will move from the blood into the extracellular fluid, and which will be able to attack the pathogen. Okay, so that's endothelial contraction, and clearly we don't really want this to occur in the terminal arterioles as much as we do in the capillaries and post-capillary venules, because if it occurs in the terminal arterioles, where's the exudate going to form? It's going to form in the vascular smooth muscle cell there that surrounds the endothelial layer. So this really occurs in the capillaries and post-capillary venules. But note that it doesn't just occur in the capillaries, it does also occur in the post-capillary venules. So these are going to contract down as well, and fluid will leave from the post-capillary venules into the extracellular fluid as well. Okay, right. So that is the second part of type 1 activation, endothelial cell contraction, which is going to result in the formation of an exudate. Now note that although fluid and proteins now can leave through these gaps are not really big enough for cells to move through yet. At least it's not big enough for them to move through unaided. They're going to require aid from the endothelial cells. And so the final thing to mention about type 1 activation is that you're also going to get leukocyteextravasation. So leukocyte is the fancy word for white blood cell, and extravasation means moving them from the bloodstream into the extracellular fluid, so taking them beyond the bloodstream, extravasating them. So leukocyte extravasation is the other thing that the endothelial cells that have undergone type 1 activation are now going to participate in, the other major change to their behaviour. Okay, now following type 1 activation, there is a specific white blood cell that the endothelial cells like to actually recruit from the bloodstream into the extracellular fluid, and this is the neutrophil. So let me just introduce you to the neutrophil. So neutrophils are, if you like, one of your weakest types of immune cells. They're an immune cell that it's quite easy to produce. They don't require that much training to produce. It's, if you like, an inexpensive to produce white blood cell, and you have a huge number of them, and therefore you can just throw them at a pathogen. Okay, and they're the first thing that is brought in to try and deal with a pathogen. So let me just introduce you then to what a neutrophil looks like. So here is its cell membrane, and they have a very characteristic nucleus. So it's very easy to identify neutrophils if you're looking at a sample of blood down the microscope, because they have a nucleus that has multiple lobes, so a multi-lobe nucleus. So they don't have multiple nuclei. Don't think that this is multiple nuclei. It's just a really odd shape for a nucleus. So the nucleus isn't a nice circular shape. Instead, it has this sort of shape where you have these multiple lobes. In this case, I've drawn 3, which is sort of like the prototypical number to draw, and they're connected by these little connections. Okay, so this is what the nucleus of a neutrophil might look like. So I'll just colour this neutrophil in yellow here. Okay, so there are loads of other names that people will use for neutrophils, and I just want you to be aware of them. So other names that people might use for neutrophils are polymorphonuclear cells, PMNs for short. So this stands for, to write it out in full, poly meaning many, morpho meaning shape, nuclear meaning pertaining to the nucleus. So these are cells that can have many different shaped nuclei, and that's actually a good point to mention. So I've drawn my nucleus as having these 3 lobes for the neutrophil, and that is the archetypal number of lobes for a neutrophil to have. However, you can have neutrophils that can have 4 lobes, or 5 lobes for their nucleus, or even 6 lobes. So there are lots of different shapes that the nucleus of a neutrophil can take. So hence why they're often referred to as polymorphonuclear cells, PMNs. So PMNs, neutrophils, they're the same type of cell, and you have a lot of these in your bloodstream. They're one of the most prevalent types of white blood cell. Okay, so type 1 activated endothelial cells are going to start participating in neutrophil recruitment, specifically neutrophils, not other types of leukocytes yet. They're just going to be recruiting neutrophils, and later on I will explain why it is just neutrophils at this stage. I will explain exactly how neutrophils are extravasated later on, but for now just know that the neutrophils can't just fit through these gaps on their own. Instead, the endothelial cells have to put all sorts of molecules on their surface to bind to all sorts of molecules on the surface of the neutrophil, and then once they've got this very powerful interaction with the neutrophil, they can sort of guide the neutrophil through the hole, and the neutrophil has to squeeze its way through. It has to become hugely deformed into like a thin sausage of a cell to squeeze through, and then it does get across the endothelium. Still again, neutrophil extravasation is something that's majorly going to be a behaviour of the capillaries and post-capillary venules, and not something that we really want to be inducing in the endothelial cells of the terminal arterioles. And because if it occurs here, the neutrophils are just going to end up in the vascular smooth muscle cell layer. So again, these two are major activities of the capillaries and post-capillary venules, whilst this one is majorly in the terminal arterioles. Although, as I say, endothelial cells of all of these different forms of the microvasculature will produce prostacyclin, which will diffuse over to the terminal arterioles. Okay, so that's type 1 activation, which is produced in the endothelial cells in response to histamine. And I'll remind you that histamine is the one that's going to be produced first and act quickest. So this is the thing that will happen within minutes of the pathogen being present within the tissue, because the histamine will be released very quickly and it will go and act on the microvasculature endothelial cells very quickly. So you'll get increase in blood flow, the formation of an exudate, and you'll start to get neutrophil extravasation within minutes. Okay, now, just before we move on, let me talk to you about the cardinal signs of inflammation. So people often talk about what are known as the cardinal signs of inflammation. So the clinical signs of an inflamed area. So these are that the area will become red, the area will become hot, the area will become swollen, and the area will become painful. So the main cardinal signs of inflammation in an area are the area becomes red, and we can understand most of these cardinal signs just from looking at type 1 activation here. So why will the area that is undergoing the acute inflammatory response become red? Well, we have increased blood flow to that area. Blood is red, so that's why the area will become red in response to the acute inflammatory response. The area will also become hot. Again, we can understand that blood is very warm, so if you've got an inflamed area of skin, for instance, that area will become red and it will become warm as well. So red and hot. Then the area will also be swollen. We can also understand that so far because we've got the formation of this inflammatory exudate, which will be leading to fluid accumulation in that area, which will therefore lead to the area becoming swollen. So that's fine. And the final one we haven't really talked about yet, although I have mentioned it. I've said that prostaglandin E2 is going to cause pain. That's the other cardinal sign of an inflamed area, but we haven't discussed that in detail yet. Now, people often love to state the cardinal signs of inflammation in Latin rather than in English, so I'll just give you the Latin words for this. So redness, rubor is the Latin word for that. Hot, calor is the Latin word for that. Calor means heat in Latin. Swollen, tumor, tumor means swelling. And then finally pain, that's dolor. Okay, so if you hear people talking about rubor, calor, tumor, dolor, those are just the Latin words for the cardinal signs of inflammation here. Okay, some people also add a fifth cardinal sign of inflammation onto here, which is loss of function, but to be honest, these are the main 4 cardinal signs of inflammation, and loss of function comes along with pain. Okay, so we can already understand the cardinal signs of inflammation then just by looking at what histamine does by causing type 1 activation of endothelial cells. What I now want to talk about is type 2 activation of endothelial cells, which is going to occur in response to tumor necrosis factor alpha and interleukin 1. So now let's talk about what TNF alpha, tumor necrosis factor alpha, and interleukin 1 are going to do. Okay, so again they are going to be acting on receptors on the basolateral surface of the endothelial cell here. Now these receptors are not going to be G protein coupled receptors, unlike the receptor for histamine. They have far more complicated intracellular signaling pathways. We didn't go through the intracellular signaling pathway for histamine. We are certainly not going to go through the intracellular signaling pathway for tumor necrosis factor alpha and interleukin 1 because they're far more complicated. This is just the classical GQ pathway. These have pathways that you will probably have never seen before. Okay, so tumor necrosis factor alpha, it's going to act on a receptor. I'm going to give you the names of the receptors, however. Tumor necrosis factor alpha will act on the receptor known as the tumor necrosis factor receptor 1, TNFR1, and I'll just colour that in in pink here. Okay, so tumor necrosis factor alpha acts on TNFR1 there, the tumor necrosis factor receptor 1, and then interleukin 1 will act on the interleukin 1 receptor type 1. Okay, so nice simple names for receptors then. And these will again both be found on the basolateral side of the endothelial cells. And these are now going to cause another activation process within the endothelial cells, but this time it's going to be called type 2 activation. So these are going to cause type 2 activation of endothelial cells. Okay, so now let's go through what's going to happen in type 2 activation. Now the first thing again to stress is that type 2 activation will occur a lot long after type 1 activation. So type 1 activation occurs within minutes. Okay, type 2 activation will occur within hours of the initial recognition of the pathogen by the sentinel cells. The reason is that the sentinel cells are going to have to get around to synthesizing the interleukin 1 and the tumor necrosis factor alpha. That will take time. Protein synthesis is not extremely rapid. It does take time. So they're going to have to synthesize tumor necrosis factor alpha and interleukin 1. The other thing is that even once you've produced those and they've acted on their receptors on the surface of the endothelial cells, the actual intracellular pathways that these two activate result in changes in gene expression in the cell. The way in which these change the behavior of the endothelial cells and cause type 2 activation is by changing gene expression within the cell. Type 1 activation didn't involve changes in gene expression. All of these things were done without having to change gene expression. The synthesis of prostaglandin I2 just involved activating enzymes that were already there. Okay? Endothelial cell contraction just involved activating proteins that were already there. Leukocyte extravasation, as we'll see later, involved putting proteins onto the surface of the endothelial cells, the apical surface of the endothelial cells, that were stored in vesicles known as viable palade bodies. I'll restate this all later on. But for now, just know that the proteins that were involved in this were already present and synthesized. They just had to be put into the apical membrane so that they could then interact with things on the surface of the neutrophils to cause neutrophil extravasation. So none of this required changes in gene expression, which meant that it was all very rapid. Whereas the way type 2 activation occurs is it results in changes in gene expression. So what actually occurs when you type 2 activate an endothelial cell? Well, you produce a huge amount of prostacyclin now. Okay, so the amount of prostacyclin the endothelial cells produce is going to go hugely up from the amount that they were producing because of the histamine stimulation. So histamine, we've talked about the fact that it does drive the synthesis of prostacyclin, but compared to what these two are going to do, it's feeble. Okay, because these two are going to drive the expression of new enzymes that will result in the production of prostacyclin I2 here. And I think I've been calling this prostacyclin prostacyclin's fine. If I've been calling this prostaglandin E2, I do apologize. I'm talking about prostaglandin I2 here. Okay, so type 2 activation, when we activate our endothelial cells with tumor necrosis factor alpha and interleukin 1, we're going to drive new expression of enzymes which will produce prostaglandin I2, so that you will get a huge amount of prostaglandin I2 produced. And this therefore again will go and diffuse to the vascular smooth muscle cells, and it will cause them to relax, and therefore you'll get vasodilatation of the terminal arterioles, leading to an increase in blood flow. So type 2 activation is just going to help this type 1 activation out. It's going to do the job better, if you like. This is the quick one that we start up right away. This is the more powerful version of it, which will go on chronically, or at least for much longer and much more powerfully. So type 2 activation is going to take over from type 1 activation. So we're going to produce much more prostacyclin. In addition, we're going to get further endothelial cell contraction, so more contraction here. And the way this is going to occur is by actually changing the cytoskeleton. So when type 1 activation changed the shape of the endothelial cells. This sort of contraction that it activated was actually very similar to the sort of contraction that you get in muscle cells. Okay, it involved proteins interacting with one another and pulling the cytoskeleton inwards. It involved contracting the cytoskeleton. What we're now going to do is we're actually going to change the structure of the cytoskeleton. So type 1 activation just involved contracting the cytoskeleton down to change the shape of the cell. Type 2 activation actually involves dismantling the cytoskeleton in the peripheral portions of the cell. So if you imagine the peripheral portions of the cell, they're full of cytoskeleton which is holding them up here. In fact, let me go back even further. I love doing this talk. So often when we draw pictures of cells, we draw this nice picture, and I'll just do it down here. You know, we draw a cell membrane and we draw a nucleus like so, and then we draw the cytoplasm empty in this way. And that's because this is a lovely picture to draw. But then what is actually holding the cell in its shape? Because the cytoplasm that we've drawn is just gloop. And the cell membrane isn't rigid, it's just gloop as well. So if this was our model of the cell, it would just continuously be changing shape, but that's not the case. Cells have a fixed shape, and the reason is that inside the cytoplasm in reality there is a spider's web, a hideous meshwork, which I would draw like this. Okay, just proteins everywhere, making up a very rigid, solid meshwork that holds the cell in its shape. And this is what the cell membrane is resting on, and that's the reason it maintains the same shape. The cytoskeleton is holding the cell in its shape and maintaining it in its shape. And you can see why we don't normally draw the cytoskeleton, because it looks like a mess now. Okay, so this is the fundamental concept of the cytoskeleton, a mature concept of the cell. So in these endothelial cells there will be a cytoskeleton, and as part of this contraction here we just contracted the cytoskeleton down, which is what occurs in muscle cells all the time to contract them. Here now what we're going to actually do is dismantle the cytoskeleton in the peripheral portions, and therefore actually change the cell's shape to open up gaps. So the cells, if you like, after type 2 activation might now look like this. They've completely lost the peripheral portions now. This maybe is a little bit extreme here, but it gets the message across. They've completely retracted their peripheral regions. They've lost the cytoskeleton there. They've dismantled it. They've changed their shape so that now there are great big gaps in between neighbouring endothelial cells, and that therefore is going to help form more exudate. So again, it's just doing the job that type 1 activation did, but doing it damn better. So that's what type 2 activation really is all about, just doing what type 1 activation did, but doing it better than type 1 activation did it. Okay, and then finally, type 2 activation is also going to result in more leukocyte extravasation. So you're going to synthesize more proteins that will help you recruit more neutrophils in. So you'll get further neutrophil recruitment, but in addition you're also going to start getting monocyte recruitment. So you're going to start recruiting a new type of white blood cell that couldn't be recruited just from type 1 activation. It required us to synthesize new proteins to actually be able to interact with proteins on this new type of white blood cell. So we're going to start getting monocyte recruitment, or you could call it monocyte extravasation. Okay, so let me just go through what monocytes are. Let me introduce you to this other very important cell of the immune system. So, monocytes are the precursors to macrophages, so you do not have macrophages circulating within the bloodstream. Instead, what you have is this type of cell known as a monocyte, which looks like this, a very large nucleus. And the reason it's called a monocyte is because it just has a nucleus with one lobe, in comparison to the neutrophils, which have a nucleus of loads of lobes. Okay, so monocytes are also very prevalent within the bloodstream. They will circulate around within the bloodstream, and what can happen is when type 2 activation has occurred to endothelial cells, and the endothelial cells will put on the apical surface a protein which they have to synthesize. So it's unlike what the type 1 activation did, where we were just taking a protein in a stored granule and putting it onto the apical membrane. We actually have to synthesize this protein now. We'll put it onto our apical surface. It'll interact with proteins on the monocyte surface, and then the monocyte can actually be recruited into the extracellular fluid, and as soon as it goes into the extracellular fluid at the site, it will then start differentiating into a macrophage, and then it takes on this much bigger blobby form here. Okay, so monocytes are the precursors to macrophages, the circulating precursors to macrophages. You know, you wouldn't want a great big blob of a cell like a macrophage here circulating in the bloodstream. It's far better to have it compact like that. Okay, so after type 2 activation has occurred, we can actually start recruiting macrophages. Type 1 activation could not activate endothelial cells to recruit macrophages. This is the reason that initially in the acute inflammatory response you firstly see loads of neutrophils appearing, and then only later on, after a few hours, do you see macrophages appearing, because you require type 2 activation of endothelial cells by the tumor necrosis factor alpha and interleukin 1. And I've just realized that this is barely visible with the current lighting, so I'll move it down to there where the camera seems to prefer the lighting that way. So only after you've had type 2 activation will you then get the endothelial cells activated enough with the correct proteins on the apical surface to recruit monocytes from the bloodstream to become macrophages in the tissue fluid. Okay, so let's just go over a summary of that then. So we've now seen the effects of histamine, tumor necrosis factor alpha, interleukin 1, and also prostaglandin I2. So histamine is going to appear first. It's the one that appears within minutes, and it's going to affect the endothelial cells very quickly. It causes type 1 activation, which will cause them to start secreting prostaglandin I2. This prostaglandin I2 will work with the prostaglandin I2 that the sentinel cells are starting to synthesize to cause arteriolar vasodilatation, which will then lead to increased blood flow to the area. That will lead to the redness and heat associated with an inflamed area. And then what we'll also get is endothelial cell contraction, and this will occur by activating the cytoskeleton to contract, and this will open up gaps between the endothelial cells and allow an inflammatory exudate to form, which will be bringing in proteins that will be able to attack the pathogen, and this will of course lead to the swelling at a site of inflammation. We're also going to start recruiting neutrophils from the bloodstream into the extracellular fluid, and these, as we'll see, will later attack the pathogen. We will go over that later on. We'll go over exactly how neutrophils attack pathogens. Then later on what's going to occur is you're finally going to get around to synthesizing tumor necrosis factor alpha and interleukin 1. Those will go and act on endothelial cells. Again, there'll be a lag whilst the endothelial cells actually undergo the change in gene expression that TNF alpha and interleukin 1 are signaling for. And then what will finally happen is known as type 2 activation, and the endothelial cells will start producing much more prostaglandin I2 because they've had the enzymes that make it upregulated, and that will cause further vasodilatation to increase blood flow further. And you'll get changes in the cytoskeleton of the endothelial cells so that they have completely changed shape, and you've got even bigger gaps between the endothelial cells, which will allow even more exudate formation, so even more swelling. And you're also going to potentiate leukocyte extravasation, so you'll put more of the proteins that can help recruit neutrophils on the apical surface of the endothelial cells, so you'll get more neutrophils recruited, and you'll also put proteins that are necessary for the recruitment of monocytes on your apical surface, and therefore you'll start getting macrophages recruited to this site of inflammation. Okay, so that is the effects of histamine, TNF alpha, interleukin 1, and prostaglandin I2. Let me now tell you the plan from here on. We're going to have a break now. We're then going to go over in brief what the effect of interleukin 6 and prostaglandin E2 is. We're then going to actually start talking about why does forming an inflammatory exudate actually help, so we'll go over the complement proteins, and then what we'll do is talk about leukocyte extravasation more. So we'll talk about the actual mechanisms by which endothelial cells recruit leukocytes so neutrophils and monocytes in this case, and then we'll talk about what the leukocytes, the neutrophils and the monocyte and the macrophages are actually going to do to help clear the pathogen, which is of course they are phagocytes. Okay, so that's the plan from here on in. So we'll have a break now.